I'm Andrew O'Brien, and my summer research project is about the synthesis of gadolinium-based high-relaxivity contrast agents for prostate cancer. A little background on this project. Prostate cancer is the second most frequently diagnosed cancer among men worldwide. Prostate cancer treatment efficacy heavily depends upon how much of the disease has spread inside the body, and contrast MRI can generate high-quality images of internal body tissue in any present tumors. This project will involve something called targeted imaging of prostate cancer. PSMA is an antigen which is found on the surface of prostate cancer tumors. DCL is a molecule which binds to an active site of PSMA. And we use this principle of targeting to increase the concentration of a contrast agent at the tumor site. In this case, we're going to use the DCL molecule and attach a contrast agent to it in order to better image prostate cancer. The targeting agent for this project will be DCL, as seen in the bottom illustration, which is made by combining glutamic acid and lysine in a urea-based pattern. Gadolinium is a paramagnetic metal, which is detectable through MRI, and it's detectable because it shortens the time for disturbed protons and water molecules to return to their equilibrium. This is known as the T1. And molecules that shorten this time are more effective contrast agents and have high relaxivity times, or R1. On the right is a diagram showing the structure of Dodoram. This is a commercially available gadolinium-based contrast agent. However, it is not targeted. If we can incorporate gadolinium into targeted contrast agents, we can selectively image prostate cancer tumors through MRI. The basic principle of this targeting is that wherever the gadolinium will be, the tumor will be too. This project will involve a modular puzzle piece-based synthesis to construct our molecules. Amino acids containing dyes and metals are coupled together with the targeting moiety added last. This allows for the targeting of different cancers with multiple different imaging agent combinations. And the diagram below shows a linear synthesis of a targeted molecular imaging agent, or TMIA for short. We have two different peptides containing different dyes or metals that are combined with the linker and linked with the targeting agent. And in this case, this synthesis describes one for prostate cancer because it's the PSMA targeting agent. Past students have created mono and di-gadolinium targeting agents. On the left is a lysine-based gadolinium targeting agent. It contains the DCL targeting group, which means that it's effective to image prostate cancer. In the middle is a digadolinium targeting agent. The gadolinium molecules are attached to lysine groups. And you'll notice that instead of DCL, there is a different targeting agent. This agent works against breast cancer cells. And on the right is a bimodal TMIA. It features two different lysine molecules. However, one of them contains a gadolinium and the other contains a dye. This is also targeted to prostate cancer, but because it contains the two different groups on the ends, in this case the dye and the metal group, this means that it can participate in different modes of imaging. And this is impressive because this functionality comes from just one molecule. This project will also investigate the use of tryptophan in TMIAs. We believe that because this spaces out the gadolinium containing lysine amino acids, increases the solubility in a variety of solvents, and is visible through spectrophotometry, that this will create more effective targeting agents. On the bottom is a diagram showing the molecular structure of tryptophan, and the aromatic group on the side chain makes it absorb at around 280 nanometers, which makes it visible through spectrophotometry, which is really useful to analyze the progression of the synthesis. The hypothesis for this project is that the use of tryptophan in gadolinium-containing TMIAs will decrease the T1 values and increase their R1 values compared to their non-tryptophan counterparts. We also believe that by creating a bigger molecule, this will also have an effect, and that will be discussed later. The goal of this project is to synthesize mono and digadolinium TMIAs, which incorporate a tryptophan spacer, evaluate the relaxivities, and compare them to existing gadolinium-based contrast agents. On the bottom is a diagram showing the names of the compounds and their corresponding moieties, or the groups of atoms inside the compound. On the left is our monogadolinium compound, and on our right is the digadolinium compound. Some key reactions in this project involve FMOC deprotection and peptide coupling. FMOC is a protecting group which is attached to a primary amine, or NH2. The purpose of this is to prevent any side reactions to the primary amine. The FMOC group can be removed through a strong base, and this enables the NH2 or amine group to participate in peptide coupling. And on the right, we have representations of the peptide coupling reactions. The first step is to use a coupling agent such as HAT2 or TSTU, and this will generate an excellent leaving group on the carboxylic acid. This will better allow for the primary amine to react with the carboxylic acid and create the peptide bond to link the amino acids. This is a diagram showing the synthesis of our monogadolinium compound. We start with deprotected lysine containing the gadolinium. The carboxylic acid is turned into an amide, and that prevents any reactions from the other end. We couple this with protected tryptophan, and we get our tryptophan spacer. We then deprotect this molecule and finally attach the DSS and DCl to create our monogadolinium compound. This is the synthesis for our digadolinium compound. It follows the same path. However, once it's deprotected, another lysine group is added on. In this case, it is the D enantiomer of lysine, and this is important because we believe this will increase the stability of the compound within the body so that it is not degraded by any enzymes. We then deprotect this compound and finally add the DSS and DCl to have our finished targeting agent. The remaining objective is to measure the relaxivities of our synthesized compounds, and we do this by measuring the relaxation rate through NMR, and we plot this against the concentration of our contrast agent, and by looking at the slopes, we can determine the relaxivity. On the top, we have the existing results for existing compounds with R1 values around 4.5. These are all monogadolinium compounds. Based on the use of tryptophan as a spacer and the effects of adding an additional lysine gadolinium group, we expect our monogadolinium compound to have an R1 value of at least 5 and our digadolinium compound to have an R1 value of at least 11. We believe that the presence of 2 lysines will create a compounding effect in which one molecule with 2 gadoliniums will have an overall higher relaxivity than 2 separate molecules with just 1 gadolinium. Future work in this project will involve synthesizing polygadolinium TMIAs through convergent synthesis. In this case, on the left side is a representation of linear synthesis, which is how the 2 gadolinium-based TMIAs were synthesized in this project. This involves one molecule undergoing a set number of steps until the final product is reached. On the right is a representation of convergent synthesis, in which many smaller molecules are brought together to create the final product. We believe that the use of convergent synthesis will increase the overall yield and decrease the number of steps per gadolinium group. This will enable us to create molecules with even higher relaxivity, and the potential for this is very high. Finally, I would like to acknowledge my research mentor, Dr. Hans Schmidhenner at the Rochester Institute of Technology. I would also like to thank the current Schmidhenner Group students for their camaraderie throughout this project. I would also like to thank the past Schmidhenner Group students for creating and teaching me many things along the way. I would also like to thank our collaborators at the Roswell Park Comprehensive Cancer Center in Buffalo. And finally, I would like to thank the RIT College of Science and the RIT Honors Program for funding for this project.